Right, so good afternoon. We are pleased to have Sir Walter Bodmer giving us a second seminar today about colon cancer genetics. His visit is being sponsored by the Storer Endowment. Today, Sir Walter is going to speak about another major research interest of him. He has done a tremendous amount of work on cancer genetics. One of the many important findings is that he discovered that APC mutations were the cause of familial adenomatous polyposis in humans. And for those of you who don't know, the APC is actually one of the most important genes in colon cancer biology. It's mutated in around 85% of all colon cancer, so that was a major findings. So today we will have Sir Walter talking about colon cancer genetics and biology. So I'll leave you to Sir Walter.
Thank you. Well, as Tracy said, my talk today is quite different. It's about our work on colorectal cancer, and in particular using cell lines. And I thought that maybe I ought to start by giving you a little bit of background about colorectal cancer, what the colon is, and then go into some of the details of what we've done, especially using the cell lines as models and why we think those are good models. And for studies of drug resistance and in relation to the properties of the tumors, and also for aspects of the study of the biology of colorectal cancer, which we believe are very important for the understanding of what goes on. Now, the colon is a tube down which waste products of the food that you eat go, and it's actually made up with large indentations at many positions, which are called crypts, and we're talking about the large intestine. And the crypts are formed by the epithelial cells, which are the cells, of course, from which all carcinomas come. And they're of different types, and the crypts are actually surrounded, you can't see this here without labeling, by a very important set of cells that are called myofibroblasts. They are cells that are of the fibroblast lineage, but they're myofibroblasts because they have properties of smooth muscle cells and play an essential role in creating the environment in which the stem cells that are at the bottom of the crypt divide and produce the differentiated cells. So the structure of what goes on is at the bottom of the crypt you have the adult tissue stem cells for the crypt, and there's good genetic evidence that at any given time each crypt is monoclonal. It's derived from a single cell, one of these stem cells that is the one that is dividing and producing by a process of intermediate cell division, so-called transit amplifying or progenitor cells, that then leads to the production of the three main types of differentiated cells that make up the colonic crypt. The columnar cells, which are the main structural cells, and that actually probably mainly extract fluid from the feces. The goblet cells, which are important, they secrete the mucus that creates an important protective surface for the colon, and the enteroendocrine cells, which are a smaller proportion of the total, that actually make endocrine functions and that are important for the function of the crypt. And this whole structure turns over in all of us every few days, 3 or 4 days. It's an enormously active turning over system, so that by the time you're my age, I mean, it's turned over many hundreds of times. And so that when you have tumors, the tumors are derived either from the stem cells or later from the transit amplifying cells, and the mutations that give rise to the cancer have to occur in the stem cells initially, because it's only those that give rise to a permanently growing cell population. The rest of these cells, when they're differentiated, they don't divide, they're sloughed off, they die by apoptosis, and are released into the lumen of the bowel. But it is possible, I think quite likely, that the ultimate coup de grace that leads you really to a cancer is a mutation that occurs in these transit amplifying cells. So that's the basic background biology. Now, it's been known for a long time, largely from work in St. Mark's Hospital in London, with which we started some of these studies, that you actually have a transition from the normal epithelium through stages that are called adenomas, sometimes polyps that are abnormal growths that are early, become larger, start penetrating the muscular layer under the epithelial cells, go further, and then eventually metastasize. And this is a progression that goes from the normal to the cancer. And in polyposis, it's called polyposis because the mutation in the heterozygous state of the APC gene gives rise to a lot of these early adenomas, only a small proportion of which eventually go on to produce carcinomas. So you can get these early stages of growth that are relatively stable for some periods of time, and of course that's the basis of screening for early cancers. And at various stages during this process, as many people, including Vogelstein and others and myself, have suggested, you get mutations, as Lewis has said, the earliest mutations in about 85% of colorectal cancers are in the APC gene. You get other mutations at different stages, all of these actually occurring before the initial metastasis, which is usually what kills a patient, because if you can get a tumor at an early stage, cutting it out, as the surgeon does, is a complete cure. It's because of the difficulty of dealing with the cells that spread and go in other places that a disease like this is hard to treat. So you get well-known mutations, KRAS, P53, mutations in the mismatch repair genes that occur. And colon cancer has been a very useful model for studying cancer as a whole, carcinomas, I have to say, partly because there are two very well-described inherited deficiencies, the familial adenomatous polyposis, which identified the APC gene, and the role of the Wnt pathway in this and ultimately in other cancers, and the mismatch repair genes, which were identified by candidate positional cloning in the 1980s. And in each of these, you have the classical Knudsen-type situation where what's inherited in the germline is a single mutation, and what happens in the tumor is the loss of the remaining wild type by loss of heterozygosity by a process of somatic recombination, mostly, as we've shown with Ian Tomlinson. And you get a similar situation here where these mutations in these genes occur in the germline and then are associated by LOH with subsequent tumors. In this case, maybe only about 15% or so of tumors that are sporadic, not familial, will have mutations in the mismatch repair genes. And you can summarize that by saying, well, there's a range of mutations that have been found in sporadic cancers, non-inherited cancers. And cancer is essentially a somatic evolutionary process where changes in that are mutations, or maybe changes in mainly in methylation, that actually are sufficiently stable to act as if they were mutations in downregulating or maybe upregulating the presence of a particular product. These are what drive, by selective advantages, the somatic evolution of a cancer. And that's why people call them driver mutations. I like to think of them as those that have the selective advantage, the Darwinian selective advantage that promotes the initial independence and then growth and spread of the cancer. So you have mutations in the Wnt pathway, in APC and beta-catenin. E-cadherin is related to this. The transcription factors, you have mutations that are often thought to be there because they promote instability in cancers. I believe that's not the case. I think they're mainly connected with apoptosis, mutations in P53, the mismatch repair proteins and checkpoints. Very important series of mutations in growth factors and their receptors. And then I put this at the end because that was my favorite study for a long time. You get quite specific mutations in the HLA class 1 genes that seem to be protecting the cancers from immune attack at the cellular level, which is what happens when you remove class 1 expression. So that's a background. I just want to say a few words about the use of cell lines. We make extensive use of cell lines, which we consider in many ways are the best models for studying human cancers. And I want to make a few points about this because there's a lot of discussion suggesting they're not good, that you should work with mice, that they don't represent what comes out of the tumors, and we believe most of that is wrong. First of all, the spectrum of mutations in cell lines follows very closely what you find in primary tumors. You very rarely, if ever, find any of these driver mutations changed. in cell lines that have been grown for a long time in culture. And there are examples of cell lines that, unknown to start with, turned out to be the same because of cross-contamination, have been grown for a long time separately and have essentially the same mutations. Very similar patterns of mRNA expression and similar patterns of methylation are found in the cell lines as in primary tumors. They don't change in culture, I've said that. Duplicates mostly show similar activity. And there are huge advantages in working with cell lines. You can do studies, functional studies, in a way that you can't do with primary tumor material. They represent what happens in the human cancer, not what you think ought to happen if you put mutations into a mouse. And you can actually, as I'll show by implication, you can reconstruct a lot of the microenvironment which people talk about by adding the relevant cells, such as myofibroblasts, in co-culture with the epithelial cells. And they are actually an advantage to study what happens in the absence of any other cells, because then you can say what you're looking at is really a function of those cells and their properties. The other thing I would want to say is we work with a panel of more than 100 cell lines from colorectal cancer. You'll see many studies that work with a few cell lines, even 20 or 30. That's not enough to look for subsets of cells that have particular properties that may go along with their biological aspects, or more specifically with response to drugs. You need enough numbers of cells to be able to be sure you get significance if you have subsets of the ones that respond. And many studies are not adequately powered in the use of cell lines. And you'll often read papers that make use of a few cell lines, which I'll mention, which are in many ways quite unrepresentative of the totality of colorectal cancers. And therefore you've got to be very careful when you're doing studies with cell lines that you do actually respect the fact that you've got to look at the variety that is occurring in the normal cancers that you see, and to see that that's represented in the cell lines that you work with. So I want to show you first some examples of one particular drug response study done by a very talented surgeon in my lab together with a postdoctoral fellow. And we were looking at cetuximab, which is a well-known monoclonal antibody against epidermal growth factor receptor, one of the key receptors for epidermal growth factor, which drives the growth of the epithelial cells. It's in the clinic, it's been widely used. And we're looking here at the resistance of cetuximab, the antibody, on its own, what its effects are on different cell lines. So these are the names of a series of different cell lines. Each of these is a response curve measured in terms of percent of the control growth after a given period of time with different concentrations of antibody. And these are a set of cell lines that are essentially totally resistant to cetuximab, to the direct effects. We have cell lines that show partial resistance. These are another set of cell lines in which you're getting some killing that doesn't go down to complete killing and goes down to intermediate levels, and are very clearly distinguished from the most sensitive cell lines, which are shown here, which actually go down to more or less complete killing by cetuximab antibody. So you can categorize the cell lines in that way. And then you can ask, well, what's the properties? What are the differences in terms of mutations, expression, biological properties between the cell lines that do and don't respond to cetuximab? And I show you here, anticipating what I'm going to say later, that we've also studied, and I'll show you some of the data on this, one of the well-known, it's actually a dual inhibitor, tyrosine kinase inhibitor, that inhibits the epidermal growth factor receptor tyrosine kinase, and also the HER2, the one that's used in the treatment of breast cancer. It's a dual inhibitor. And this shows that the results you get actually with the antibody and with this low molecular weight dual inhibitor are very highly correlated, because that's the direct inhibition with cetuximab, and that's the effect of different concentrations of lapatinib, and you get a really remarkably good correlation. So what are the data then that relate the response of cetuximab? What we're plotting here is we're plotting the percent of control growth that's still there. So that's the level of growth inhibition, I should say. So these are completely resistant, getting up to a bit more resistant, more resistant at an intermediate level, and these are the sensitive. So these are all names of different cell lines that we work with. They're ordered by the level of response, going from the most resistant to the most sensitive. And what you see up here in each line is the presence or absence of mutations in particular genes that are well known to be present in colorectal cancer. And KRAS has been much described, and in fact you can't give cetuximab now to patients unless they've been evaluated for KRAS mutation and shown to be wild type. So where you've got a filled in yellow symbol here, that means those are mutant cell lines. Where it's not filled in, they're wild type. That's for BRAF, which is one of the members of the family, of the RAS family, related family, and that's for a kinase that's very important in these pathways. And these blue and the yellow distinguish different types of mutations, some in one exon and some in exon 20. Now, what you can see here, just looking at it, that the most resistant, and we're just arbitrarily dividing these into 3 groups in terms of level of resistance, to be more or less equal in terms of the extent of their resistance. And you see that all the sensitive cell lines are wild type for all these genes, ignoring the fact that these are mutations in exon 9, because there's a lot of evidence that exon 9 mutations are not as functionally relevant as those in exon 20. And as you move down to the more resistant cell lines, you get more and more expression of the RAS mutants, which you expect, but also these others. And if you do the proper statistics of that and classify resistant, partially resistant, and sensitive, presence of one or more of these mutations, this is 34, 8, and 0 there, presence of wild type is there, and if you do a statistical analysis of that, even with these numbers, you get huge significance. So there's an enormous effect of whether you're wild type for these genes on whether you respond to cetuximab. And that correlates with what's subsequently been found clinically. In other words, the effects of these other mutations have been shown to be present in clinical data and extend what needs to be done in order to be sure of reasonably getting a better result with cetuximab treatment, in addition to looking for KRAS mutations. Now, I've mentioned already that we've got similar data in lapatinib. I'll just show you that to show how similar it is, plotted in a different way. So here, this is percent kill. So the more resistant ones have less killing, more killing, and most killing if they're sensitive. The same sort of picture with respect to the mutations, and the same sort of data here showing very strong significance with having only wild type in those genes. But you will notice, before you ask me the question, what about the cases where there's wild type in all these genes, but they're actually still resistant? So there's no doubt that these mutations don't explain the whole pattern of response to cetuximab. So one way that we've looked at this is we've said to take the cell lines that are all wild type in this sense and compare expression levels between those that are sensitive and those that are resistant, and look to see if there are some obvious differences in the expression of genes between those two that might account for some of the difference as to which are resistant, which are sensitive. And these data show the results here. So again, these are all different cell lines. You can see that if they're in purple, those are sensitive. The green are the resistant. These are the partial responders. And there were 3 genes that were at the top of the list in terms of level of expression. So low level is in blue, high level is in pink, and you just look down here and you can see that the ones that are high level of expression for all these 3 genes are all resistant or partial responders, suggesting that that is a property that should indicate amongst the multiple wild-type ones which ones are likely to respond to cetuximab or not. And in fact, it's mainly DOC4 that seems to determine this, because you'll see that the high levels here are all associated either with resistance or partial resistance. So that suggests, we haven't had time to follow that up, that maybe the level of this DOC4 is relevant, and now one can begin to do studies knocking it out, for example, to see whether that influences the level of response to cetuximab. And this shows the sort of thing that you can do if you've got an adequately powered cell line panel to do this sort of work. Now, so far, I've been talking only about the direct effects of cetuximab. And I'm sure many of you are aware that an antibody response may depend on immune function, so that there are immune cells which will kill a target if there is an antibody attached to the target. Notably, NK cells, which are present in the normal blood circulation, so that if you add immune effector cells with antibody, you should get killing, but in the absence of the antibody, you would not get killing. In the absence of the immune effector cells, you would not get killing. And you can set these assays up in vitro, and in a way, as I've explained, that's a way of recreating the environment around the tumor that matters, in this case, with respect to response to the immune effects of antibody. Now, if you do these studies, then you find that there are differences in the effect. So here you're looking at the concentration of antibody, different concentrations, percent cytotoxicity, different cell lines, increased killing in the presence of increased amount of antibody, but all of this done in the presence of a source of effector cells, essentially peripheral blood cells, which contain the NK cells. And these are cell lines that are expressing the epidermal growth factor receptor at a high level. These are cell lines that don't express it at a detectable level at all, and you see they're not killed. I mean, that's reassuring because it means the assay is actually working. And these are cell lines at an intermediate level of expression, which are killed to a reasonable extent, but less effectively than the cells which have higher levels. So in this context, the response to the immune-mediated killing of the antibody is a function of the surface concentration of the epidermal growth factor receptor that is the target of the monoclonal antibody. And actually, although I didn't show you the data, the use of the direct effects of the antibody or the inhibitors of the tyrosine kinase activity associated with the epidermal growth factor receptor, that response is not significantly dependent on the level of surface expression of epidermal growth factor receptor. And that's an important result, and it actually, in addition, one can show that the killing by antibody, by immune-mediated effects, is not influenced at all by whether they're mutant or not for these genes. That's an important observation from a clinical point of view, because it says that the antibody killing, if you can make it work effectively, can override the limitation of the relatively small proportion of tumors that have the appropriate genetic makeup to be killed by the direct effects of the antibody. And there are now very powerful techniques for improving the killing effects of antibody by immune mechanisms. And so it's becoming very important to get these into the clinic and then to be able to treat patients without having to take note of anything other than the level of epidermal growth factor receptor. So I believe that that shows the way in which cell lines can be used very effectively for studying the conditions of drug response in a very direct and clinically relevant way, and ways that we know from a fair amount of the data that's already been obtained that the results in the clinic do actually match very well what we see in cell culture. And there are other examples, many of the most interesting examples, in fact, of novel treatments that work or novel treatments that are suggested to work have come from studies of cell lines in these ways. Now I want to switch completely to a different aspect of the topic, but which I hope you'll see is relevant to what I've just been talking about. That's to try and understand more about the biology of colorectal tumors. Hanahan and Weinberg wrote a widely reported perspective on what they think are the major things that are involved in getting tumor evolution and progression, and they missed out, to my mind, the most critical aspect, which is the control of differentiation. It's been known for years from pathologists that actually a poorly differentiated tumor is much more dangerous, much more likely to give you an aggressive tumor and metastasize. And it's blocking differentiation, which is one of the key steps in the development of many, if not most, cancers. So we've spent quite a lot of time working out how we can study the control of differentiation in vitro using cell lines. And this started with a very talented pathologist who did his PhD with me, Paul Richmond, many years ago, 25 years ago. And we were looking at ways of studying differentiation in culture, and people had suggested throwing all sorts of things at cell lines that later we found didn't really differentiate very much. And what he found, in particular starting with the cell line, which we have worked with a lot, but which is just one of several, that when he grew this cell line in matrigel, in a three-dimensional gel system which provided some of the extracellular matrix components that separate the epithelial cells from the outer fibroblast layer, which are key to the control of their differentiation, that this cell line would produce colonies that really looked a little bit like crypts. These are direct pictures of these colonies grown in matrigel, which is a readily obtainable source of a mixture of extracellular components. And you can see these individual epithelial cells organized in a polarized manner around what we call a lumen. And actually it took us a long time, longer than it should have taken perhaps, to go back to these studies and to look at if we could understand the nature of the differentiation process. And Trevor Ewing, a surgeon, colorectal surgeon in my lab, progressed this further, and he put it in the terms that some of these colonies you get in matrigel are large, differentiated colonies, but you also get some small colonies that look as though they're not going anywhere. And the question was how to distinguish that heterogeneity within the cell line. A cell line is not like a chemical reagent. A cell line has heterogeneity, much of which is not connected with genetic heterogeneity, but actually with the propensity to differentiate. So if you take this particular cell line and you subclone it in matrigel, you can take the cells from the big differentiating colonies and replate those. And if you do that, they reform some of the large colonies and some of the small colonies. If you take the cells from the small colonies, they can't reproduce. They just give some small colonies and none of the large differentiated colonies. And if you take a relatively small number of cells from these big differentiating colonies and put them into immunodeprived mice, they give tumors relatively quite efficiently. You'll mostly see people using millions of cells or 500,000 cells for getting xenografts. This works quite well with 200. If you take 1,000 cells from the small colonies, you get no tumors. So these cells are potentially what one would call the stem cell. There has to be a cell within the culture that's driving the growth of the culture just as it drives the growth of the tumor and throws off differentiated cells which don't contribute to the subsequent growth of the tumor. And that's the whole notion of the cancer stem cell, which is a fundamental notion for understanding the nature of the growth of a cancer. And apart from anything, if you don't treat the cancer stem cell and kill it off, you'll never get rid of the cancer. So that's one step towards identifying within these cultures by this functional assay which are the stem cells, that the stem cells are the one that can give rise to these colonies. If we take the ability to form lumens as a measure of the ability of a cell line to do some differentiation, you can show that different cell lines vary considerably in this ability. And Sean Gandhi, another graduate student, he did a survey of a range of our cell lines, and you could classify them fairly easily into those that form lumen structures and those that don't. Now, many of you, if you worked with some colorectal cell lines, you'll recognize HCT116 widely used, TLD1 widely used. These are non-differentiating cell lines, and yet people still use them to try and study differentiation. But you'll also notice that some cell lines, HT29 is another one, which I'll talk about more later, actually make relatively few and sometimes hard to distinguish lumens. So that these are not all equivalent. They have variation, but they do have the property that they can at least differentiate to some extent. So there's considerable variation in this property, And we can ask, what is that variation associated with? What are the genes that drive it? Do they influence the response, for example, to different drugs? Can one find drugs that interfere with the differentiation process in the sense of inducing it more so that you can stop the growth of these more aggressive tumors? So this comes to the discussion of a particular homeobox gene transcription factor that we've studied a great deal because of its importance in the control of differentiation of the colorectal epithelial cell. It's one of a class of 4 similar genes that you find in humans and in mice. They come from caudal, initially from Drosophila, and CDX1 and its partner CDX2, which we haven't studied to the same extent, are genes that are known to be key to the differentiation of colonic epithelial cells. They do function in the embryonic intestinal environment. In the adult, they're restricted to the small and the large intestine. They regulate some well-known genes that are intestine-specific, notably Carol Chan, who did that in my lab, showed that they directly control the expression of keratin 20, widely used to measure extent of differentiation in colorectal tissue. And interestingly, they have often been shown to be downregulated by methylation in the promoter region in tumors, something we've studied quite extensively, and I'll talk about in a moment. And there's another phenomenon that I won't say much more about, a very interesting phenomenon, that you get the gastric tumors, stomach tumors, arise from areas that are called areas of intestinal metaplasia, which start off by parts of the mucosa of the stomach looking like the intestine, expressing intestine-specific markers. It's a very interesting phenomenon of transdifferentiation, which we think is in part connected with the control of expression of this gene. And there's a similar thing involved in what's called Barrett's metaplasia, which is the origin of most esophageal adenocarcinomas, a rapidly increasing tumor in humans. Now, if you look at the expression of CDX1, you find that the expression is generally limited to the upper part of the crypt. You can see a very clear crypt going down there here, no staining at the bottom. These different crypts are obtained at different angles. And if you look at the keratin 20 expression, you can see that quite similarly you get very strong expression in these upper parts where you've gone from the stem cell through some of the progenitor cell stages to some of the differentiated stages. If we look at the methylation patterns, this is just a crude measure of the number of CPGs in the promoter region of the gene, defined essentially by homology between what you find in the sequence 5' upstream of the start point in humans and mice. Then you can classify those in which you've got significant CPG methylation and those in which you haven't, and that's what's here. So these are low levels, those are higher levels, and you can correlate that with the message level of expression, and here you can see that the ones that are not methylated are the ones that express, and the ones that are methylated are the ones that don't express the message, and you can show that that expression correlates absolutely with protein expression by using appropriate monoclonal antibodies. So while one can argue about the role of methylation in different situations, I believe that this is one of the clearest situations where the normal, mature colonic epithelial cell will not have the CPG islands and CDX methylated and will express CDX1. The change is that you have the methylation and you suppress the expression of CDX1. This is a selected subset which is enriched for those that have done that. And that makes sense in a way if you think of it in terms of down-regulating the expression of a gene that controls differentiation and therefore contributes to the blocking of differentiation. So I think this is actually one of the clearest examples of selection for methylation that is quite easy to explain in terms of its effect in terms of gene function. And while you get occasional mutations in CDX1, they're quite rare, And essentially all of this difference is explained by methylation. Now, we've looked at the patterns of methylation on our cell lines in relation to differentiation and in different ways. The first thing is you can take the dense lines, which are the ones that don't differentiate, the lumen-producing cell lines, and simply ask if you classify for high and low levels of CDX1, what happens, and you get a hugely significant correlation with whether or not they differentiate, which fits in with what we're expecting. If you take a cell line like HCT116, which doesn't form lumens, or DLD1, which doesn't, and put in CDX1 by transfection, the cDNA, then you start getting formation of lumens, which really shows that that process is to a large extent at least dependent on CDX1, even if not that's not the only factor required. And you also get a decrease in clonogenicity. The same is true for this cell line, and this cell line which forms some lumens. Anyway, when you put in CDX1, the proportion of cells that form lumens is higher and the clonogenicity is lower. So we believe that that really shows that we have got evidence that CDX1 is controlling the expression of the differentiation of those anthracyte epithelial cells. Now, there are many ways in which you can try and enrich for stem cells in any sorts of tumor cells, including a cell line, and there are many approaches being used, and one of those that's been reported is to use antibodies to the determinants CD44 and CD24, so that if you pick cells from a culture which express CD44 and 24 at high levels, you seem to enrich for colonic stem cells in the sense of which I've described it, and we've done that, but I want to show you first just something that illustrates, I think, that we are really looking at stem cells in these cultures. So you can take a single cell that's come from an enrichment by this process and look at the colony that it forms in matrigel. So this is the colony, the same colony studied with different antibodies, single colony from a single cell in matrigel. You label it with EpCAM, which some people say is differential for stem cells. We find that this is an antibody we made many years ago. It's very good, and it stains essentially all the cells, and it's a membrane antibody. This is an antibody we made against CDX1, and it stains these cells quite well, and it particularly stains them in this periphery, suggesting that these are really the differentiating enterocytes. You can stain for chromogranin, which is a product of the endocrine cells, and that's made in the same colony. And you can stain for the presence of mucin, and that's made in the same colony. So this one colony from a single cell line can make each of the three differentiated types of cells. The enterocytes keep switching off. What's happened here? Can anybody help me on this? Well, it's coming back now. I've got to go back. Okay. It doesn't like the fact that I spend too long talking about each slide. So that single cell can make all those three sorts. That's the type of cell that gives you tumors. That single cell is clearly a stem cell as defined in the cell line. Now, we've done a lot of work characterizing those lumens. This is work done by another postdoc in my lab, Neil Ashley. And to start with, he's found, and that's generally been observed before, that if you stain for filamentous actin, and here you're looking at the staining for filamentous actin in normal crypts. This is the normal colon, and the DAPI is staining the nuclei of the epithelial cells. These are crypts here. The F-actin is staining the inner surface of the lumen, where you get this filamentous actin. And this is looking at longitudinally along a crypt. This is cutting across, and there you can see this is the smooth muscle layer that underlies the epithelial cells, and you can see the staining by F-actin. So this suggests that one of the ways of characterizing maybe more objectively the presence of a lumen is to stain for F-actin. And that is in fact the case. So these are different cell lines. I mentioned that one is what W122, another widely used cell line. And here are these cell lines that don't really make lumens. And you can see this is staining with F-actin. and DAPI for the nucleus and overlay, you can see that in these lumen-forming cell lines, you get these clearly defined structures in the middle of the lumen, which you expect they're actually representing the brush border. And while you get some staining with F-actin in these other cell lines, it's disorganized. And actually you can set up automation to distinguish whether you're producing these sorts of colonies or these quite easily, which we can use then for testing drug responses with respect to whether they influence differentiation or not. Now you can use a range of other antibodies to say, well, what are the other products that are expressed by these lumens? And basically they're antibodies that detect proteins that are associated with the brush border. And one example, a widely used identifier of brush border and differentiated epithelium is vilin, and you can see when you stain for vilin and actin and overlay, you get a beautiful overlay of the staining of vilin and actin. Now what you can see here is this cell line which doesn't form lumens properly, doesn't differentiate properly, but it does express vilin. This shows you can't use just one cell marker to identify whether you've got differentiation or not. The differentiation is a complex process of producing polarized cells that form those structures that depend on, of course, the surface interaction between the cells. And furthermore, you can see that these two cell lines don't express vilin, and that's because they don't express CDX1. And in experiments similar to the ones we've done, it's been shown by others that actually vilin's expression is controlled by CDX1. So that fits together completely there and shows the sorts of things that you can begin to identify and ask, what's the difference between this cell line that enables it to express vilin but not to differentiate properly, even though it's also expressing CDX1. And just one other example, which I show because I think they're such pretty pictures. Here you've got an example of eslin, which is another important component of the, what's going on here? Someone? There we are. Okay. It's another important component of the brush border, and you can see how you get complete overlay of the eslin staining with the F-actin staining. And again, in another cell line, which I've mentioned, you get staining for eslin, but it's totally disorganized. So this cell line, which doesn't differentiate to form lumens, nevertheless expresses one of the characteristic differentiation markers of these cells. And it's something to do with the organization, the polarization control, that actually gives rise to the proper differentiation. Well, I mentioned, I won't go into the detail here, that you can purify by looking at high levels of both CD44 here and 24, this effects diagram. You're picking out the extremes of high level of both these antibody reactions. And you can show that when you take the ones that are both high, you enrich for the lumen-forming cells compared to the others, and that's true in these two cell lines. And we can use that as a way of enriching for these presumed, which we believe to be the cancer stem cells. I want to just say a little bit about this stage. I know time's running on, about the effects of hypoxia. Hypoxia is a widely discussed and important phenomenon. Low oxygen concentration in tumors associated with resistance, for example, to radiation. And you can study this in cell culture. You can grow the cell lines like this one in hypoxia, and when you grow them in hypoxia, you actually block the lumen formation, you block the expression of CDX1 and of the differentiation marker for goblet cells. That's the same in two of both of these cell lines. You don't see any of these goblet cells here. You do see some increased expression of other proteins that are associated with the maintenance of the stem cell state, which I'm not going to have time to talk more about. BMI1 is one of the polycomb factors, and Notch1. Now, there's a convenient way to study this, because it's known that the hypoxia sensing mechanism involves enzymes that hydroxylate lysines in what's called HIF1, the hypoxia-inducing factor. And there's a well-known inhibitor of these proline hydroxylases, and you can use that as a surrogate for hypoxia, which is very convenient. So if you grow the cell lines in matrigel, in the presence of DMOG, then actually what happens is you block, you see in this case, you completely block the expression of CDX1. DMOG blocks the expression of CDX1, which is what you expect. It's associated with the fact that it blocks the differentiation of the enterocytes. It doesn't block the expression of cytokinin 20, which is controlled by this, and that's a function of turnover. So the CDX1 as a transcription factor is turning over much more quickly than the structural protein, the cytokeratin 20. So you can remove CDX1 but still have a residual expression of cytokeratin 20. And you can also show what I've already mentioned, is if you use this DMOG as a surrogate for hypoxia, which incidentally shows that it's HIF1 or one of the HIF enzymes that is dependent on prolyl hydroxylase to function and to be stabilized. If you block that so you stabilize them, then you also block the expression of the goblet cell mucin. This is an antibody we have against goblet cell mucins, and when you look at the cell lines going in DMOG, they don't express it. Actually, what you can come up with then is a sort of generalized picture of what's happening in the stem cell differentiation pathway, which is somewhat similar to whether you're thinking about the normal tissue or the tumor tissue. You've got a stem cell, progenitor cells. You've got a bifurcation of the differentiation of the progenitor cells into the columnar epithelial cells, which is controlled by CDX1, and into secretory lineages, which include the enteroendocrine cells and the goblet cells, where the main control seems to be by CDX2. That's work that others have done, which we're looking into. And there's an involvement of Notch in this, because when you activate Notch, you actually prevent this process of differentiation. So I hope to show you by that how we can study these differentiation processes in culture, ask how they relate to drug responses, and consider ways in which we could use products that induce differentiation, or help to induce it, an old idea, as another way of approaching the treatment of colorectal cancer. Just in the last few minutes, I want to tell you about some more recent work, again done in my lab by Neil Ashley, that shows that the techniques now available for growing cells from primary tumors are hugely improved. In the past, maybe no more than 10 or 15% of tumors, of say colorectal tumors, would give rise to cell lines. We can now get reasonable growth from 50 to 80% or more of any fresh tumor material. And just very briefly, it does involve rather special conditions. Neil finds that you've got to disrupt mechanically rather gently. You've got to actually isolate structures that look about crypts, grow them initially on a non-attached cell surface, which actually promotes the lack of differentiation before you start propagating them on an attached surface. And he's had good results with this. Just to show you a few examples of what you get. If you do this isolation procedure from normal material, you're more or less isolating whole crypts. And you get crypt-like structures from tumors that you isolate here, and you can see here. And then when you grow them, they can eventually form spheroids. Many people would call these organoids. And you can look to see which of these have got DNA in, and you find a lot of debris that's produced by these that has actually dead material pinched off from a dying cell. And this is just a picture of a growing colony at different times. And more recently, Neil's got very nice time-lapse photography where you can show a single cell that gradually grows out to become a lumen-forming colony. And just another example then of the relationship between what you can see in culture and what you find in the tumor. In these cases, you've got the primary growth of the tumor. You can look at the paraffin blocks of the tumor that have been looked at by the pathologist. And here's using an antibody that stains for goblet cells, and simply showing that when you find goblet cells in the primary, which a lot of the time you don't find, you also find it in the parental tumor. So you're matching what you see in culture with what's seen in the tumor. And when you don't find it there, you don't find it there. You find low levels. And you find quite a nice correlation between the presence of glands that have the goblet cells and make them mucin, as you see them in vitro in culture, and from what you see in the actual tumor when it's looked at by the pathologist in a paraffin block section. And finally, just to show another example of that, if you take a tumor, this is a tumor, this is the suspension culture of this tumor. That's the paraffin block section showing that you can see there these differentiated structures. That's what happens when you grow them out on a non-attached cell. You don't see that if you take them from a high-grade, non-differentiated tumors. And then if you put these into matrigel, you get these lumen-forming structures. So it just remains for me to thank all those people who've really done the work. I mentioned Trevor, Sean, Jenny Wilding's a senior postdoc in my lab. Neil has done a lot of the latest staining and the very elegant work on in vitro outgrowth. Shaz and Jamila and then Angela before them did some of the drug response, and we get help with others. So I hope I've convinced you that you can do an awful lot of very interesting and useful work using cell lines, in this case from colorectal cancer models, with direct relevance to what happens in the clinic. Thank you. Yes.
So in the first part of your talk, you gave a very elegant description of all these different kinds of cell lines from colorectal cancer samples, and able to correlate a response to, say, an antibody, and looking at the specific genetics in those cells. And my question is, in a tumor that's in a person, would we expect that tumor to be derived from one of these cells and therefore be very clonal, and from this information it could inform us how to treat it? Or would we expect that a cancer in a person might be very heterogeneous, and therefore our treatment might work on some of the cells but not others of the cell?
That's an important question, hugely discussed, I think often in a confusing way in the literature. And I start with the answer. If you look at any evolving process at any given time, you're never going to see only one component of the evolution. If you look at whether you're thinking of it as organismal evolution or evolution in a cell line, you don't immediately get the most advantageous cell replacing everything that was there before. So that at any given time you would expect to see there may be independent lineages that have derived from a common source or different stages of an evolutionary process. So you would expect to see some heterogeneity. But the evidence is, as one would entirely expect, that it's hierarchical. You start off with one cell, and then you get a mutation that gives an advantage. Then you may get another mutation that gives an advantage, and you still see the previous one. Then you may get another mutation that gives a slightly different advantage than another one. So you're bound to see a measure of heterogeneity. It's in the nature of things that you would. What the heterogeneity that matters is that with respect to the changes that are really driving the tumor, that have the strongest selective advantage. Every time a cell divides, you produce up to a dozen mutations. There's always enormous heterogeneity. Every cell in your body, actually, that's a stem cell is different from every other cell, and is different from the cells that you grew up with, the initial fertilized cell, because there are mutations all the time. But what matters is whether those are functionally relevant. So that's my answer to the question of heterogeneity. How you deal with that is, if you've got a tumor, then you can ask the question, what are the components of that tumor in this context? So if you do deep sequencing, it's very clear. You can look for how many clones there are. Usually, and that's been done, it's been published. Swanton's written extensive papers on this, I think exaggerating the importance of it. And you would expect to find a few examples of clones in there. And each of those clones is just like an independent cancer that I'm looking at. So if you're going to treat that cancer, you've got to treat it as if you're treating every one of the single different clones. But that's not going to be hundreds. That may be 2 or 3 or 4. But you've still got to have this information on what the response of each clone is likely to be. So that would be my response to that. I think it's a problem, but it's a soluble problem, especially with modern technologies of how to deal with it. Yes?
It's a follow-up question, really. So because you've gotten better at culturing hierarchy
Yes.
now, can you run some of those same experiments you showed us at the beginning? Ask us what's the
Yes. That's a very important question, and there's a lot of interest in that. And the answer is yes. I mean, we've begun to show, Neil's done this very nicely, it's amazing what you can see just by looking. Right? So you treat one of these cultures, say with oxaliplatin or one of these drugs, and you can actually watch it killing the cells over a period of a few days. And you can do that with very few cells. So my guess is you'll be able to take primary cultures over a short period of time and assess to what extent a limited number of the drugs that you think might be best for treating that patient, do they actually treat that cancer? And I think that's going to be an important and useful development. But it depends on knowing what are the likely properties that are going to go with the treatment you want to use. Because the extent to which you can do that, we can do these sorts of assays with effort, because the cell line's unlimited, on large numbers of drugs, large numbers of combinations and many different concentrations to try and find out what's the best concentration and combination that works for a given type of tumor. And you need that information to decide, well, which, having got that information on your tumor, may be heterogeneous, that suggests that these are the drugs we ought to use. Then you can say, well, are they really going to work on that tumor? And I think that will be important. And I think the way to do it is with primary cultures, not using immunodeprived mice, which is widely promoted. Yeah.
That would be exactly my question. When you make the case for women's cell lines,
Yeah.
and in particular here on campus with Jackson West, there's considerable effort to do xenografts, patient-derived xenografts in immunocompromised mice as a model for evaluating treatment down to the individual patient. And I was wondering what your concerns are on...
I'll tell you what my concerns are about that, and I would not support it at all. It might have some role. First of all, I think we can do it a lot more effective, cheaper, and quicker than you'll ever do it with mice. There's no point in using mice if you don't need to. I think xenografts are not good models. They don't give you the environment in which you find a human tumor. They cross species incompatibilities. I just think they're very poor models of a human tumor. And you can get very good models of human tumors if you construct them in the right way in mice, essentially using the mouse as its own model, looking at the functional mutations, the sort of work that, for example, Calvin Kuro has done at Stanford, and also, of course, the people, Hans Clevers and his group. And that's different, and you're studying the particular functions of particular genes. But I think if you're studying drug response, I think it's verging on the unethical to say use mice for this. Yes?
One where your opinion about the cell line being representative of the original tumor. I mean, you said that the original tumor is made of a heterogeneous group, and when you're putting something in culture over a course of passages, 20, 50, 100 passages, aren't you going to be having
You're expressing the prejudice that I try to argue against, which I get all the time. First of all, I've shown you, and there's a lot of data, we just did a review on this, that actually what you find in the cell cultures mimics what you get in the tumors very well. There's no evidence that you get strong selective changes when you keep growing things in culture. Why one should think you get any less selection when you put them into new mice, I don't know. It seems totally and utterly irrational. The question of heterogeneity, I've already answered. You get that heterogeneity, and there's strong evidence for it, even from xenografts, whether you're looking at xenografts or cell cultures. The only way to deal with that is to teach each component that really matters of the heterogeneity as if it's a different tumor and account for that in what you're doing. I can see no case for arguing that you get anything better from looking at xenografts than you do from looking at cell lines.
I'm not suggesting xenografts are better. My concern is how good are the cell lines when you're going to give a selective advantage to one of the heterogeneous components of the tumor.
We're not. What I argued for is that if you know what the components of the heterogeneous tumors are, then they should be represented amongst the cell lines if you've got a sufficient panel of cell lines to be representative of a variety of colon tumors. Because yes, it may be true, and probably is true, whether it's a xenograft or a cell line, that actually what you grow out in culture is only one of the different components. But there are examples, and we've got them, and they're well known, where people have grown out cell lines from the same tumor in different ways and got different things. So this heterogeneity is not some sort of mysterious thing that you have to treat as though you don't know anything about it. It's simply as if you were looking at a mixture of different cell lines of given properties. And I don't think it's any different than that, and I think you have to take that into account in establishing the real properties of the tumors. And I think probably the easiest way to do that is by very early deep sequencing, because that very quickly shows you what the nature of the subclones that you've got. And then you can match those up to what you've got in your cell lines and say, what's the right mixture to treat that tumor? That would be my view of what one can do, and I think do much more effectively and efficiently using cell lines. And, you know, to study the range of drugs and drug combinations and concentrations in xenografts, even if they were good or better than the cell lines, would be impossibly expensive in terms of animals and costs. You'll never do that. And you can reconstruct, in my view, a lot of the aspects of the microenvironment. I haven't gone into the details, adding myofibroblasts, you can add immune cells. You can look at the effects of that secondarily to what you first find out by looking at the tumor cells themselves. Oh, there was another question somewhere. Yeah.
Also, going back to the other part of the talk, in your assay with cetuximab, so how does that work? How does it inhibit the growth of the tumor?
Well, the assumption is, and that's been the assumption in using Herceptin as well, is that actually the way the antibody works is by blocking the receptor. So you're blocking the access of the epidermal growth factor to the receptor. Well, every bit of serum, most of these things are just done in serum. Serum contains all the growth factors that you need for routine growth of these cell lines. You have to be careful when you grow out these primary tumors, you don't use serum. Use defined media, more or less defined. Sometimes the companies don't tell you what's in them, but they're defined essentially in terms of the growth factors, and that's very important because you may get a mixture of things that induce differentiation from things that induce growth. But in terms of what we were doing here, there's enough EGF there, and you can control that. You can go that. You can look at them, you know, for a while without EGF and then see what happens. If you add EGF, you can show that you're blocking EGF function because you can use antibodies to the phosphorylation of the receptor, and so you can do all those things, which I didn't have time to go into. But the thing about that blocking is antibodies don't easily get to tumors. And actually, in contrast to what you might think, you'll block more easily the lower the level of growth factor receptor. Because the higher the level is, the less likely you are to saturate all the available growth factors, molecules on the surface. So that's why I think you get this subset only of responses. And if you look clinically, it's quite clear that there's probably a mixture, and I think we haven't learnt yet in the clinic, and that's happening now by producing modified antibodies to get much more effective immune-mediated effects. In the lymphomas and leukemias where you've got rituximab, you've got a much easier problem because you've got very direct access of the antibody to the cells in the blood. Yeah.
Different genetic pathways associated with different prognosis, i.e., for instance, MSI positive tumors have much better prognosis than CIS positive tumors. And that has been also seen in these new 4 key cancers, that's in patients with 4 key positive cancer tend to have better, better survival. So, how do you think that this
Well, the first question you had, the mismatch of defective tumors, we actually suggested as soon as those ideas came out that one very plausible mechanism there, I don't say it's necessarily correct, is connected with immune response. And in fact, because the mutational process is producing a lot of potential targets for immune response, there's been some very nice work showing specific T-cell responses, for example, to the TGF-beta2R mutations you get in them. That's going to limit more, to some extent, the ability of those tumors to grow. So I think that's one possible reason. We have shown, though, and I don't say that this is explained, the mismatch, there's an effect on 5FU response of mismatch repair defective tumors, and I think it goes down that if they're mismatch repair defective, you don't get the same effectiveness of 5FU. I always forget which way around it goes. And so there are other effects. I'm not saying that's the only one. But you know, mismatch repair defective, my senior postdoc, Jenny Wilding, did a beautiful analysis of the expression changes in mismatch repair defective tumors. Thousands of genes are changed in expression. But there's one category that's very interesting, mostly, and they're ones because you've got repeat sequences, A and T repeat sequences in the 3' region, which are recognized by proteins that control the turnover of the message. You actually get substantial effects on message turnover in mismatch repair defective tumors, some of which may have functional consequences that are simply a byproduct of that mutational process. And that's been studied quite a lot in the context of protein sequences, of exonic sequences. But actually, in terms of expression levels, the major effect seems to be there. So there are all sorts of side effects you could expect from the existence of a mismatch repair defect in the cell line.